Meiosis I and Meiosis II by Ubaid Al-Ani and Kareem Yusuf. Meiosis is a form of cell division that halves the number of chromosomes when forming specialized reproductive cells, such as gametes. Meiosis involves two divisions of the nucleus, Meiosis I and Meiosis II. The stages of Meiosis I are as follows, starting with prophase I. The chromosomes condense and the nuclear envelope breaks down. Homologous chromosomes pair along their length and then crossing over occurs. Metaphase I. The pair of homologous chromosomes are moved by the spindle to the equator of the cell. The homologous chromosomes, each made up of two chromatids, remain together. Anaphase I. The homologous chromosomes separate, as in mitosis. The chromosomes of each pair are pulled to opposite poles of the cell by spindle fibers. But in meiosis, the chromatids do not separate at their centromeres. Finally, we have telophase I. Individual chromosomes gather at each of the poles. The cytoplasm divides, forming two new cells. Now let's talk about meiosis II. Starting with prophase II. A new spindle forms around the chromosomes, as shown. Then, in metaphase II, the chromosomes line up along the equator, attached at their centromeres to the spindle fibers. Next, in anaphase II, the centromeres divide and the chromatids, now called chromosomes, move to opposite poles of the cell. And finally, in telophase II, a nuclear envelope forms around each set of chromosomes. The spindle breaks down and the cell undergoes cytokinesis. The result of meiosis is 4 haploid cells. Thank you very much for watching.